Hello guys, today I'm going to be talking about amino acids and ways to identify them, how can we tell from non-polar to polar and acidic to basic, and how to identify them basically. And at the end, I'll be talking about proteins. So before we begin, I'm going to go over some basic definitions. So what does non-polar mean? What does polar mean? Non-polar just means it has no charges, it's neutral. While polar means it has a charge, it either could be positive or negative. So with that out of the way, let's begin on the left side, focusing on those R groups. R groups can be anything that's highlighted green. So what we see here is we see a trend among the R groups, and we see that all these R groups have hydrocarbons, and the reason they all have hydrocarbons is because hydrocarbons don't have any charges; they're considered neutral. So going off of that, a tip I made for this was that if the R groups just have pure hydrocarbons, it's considered neutral because hydrocarbons can't have a charge at all, and it's going to be a neutral amino acid, so it'll be non-polar. So going off of that tip, let's identify some non-polar amino acids. So this would be non-polar, 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 this one, this one, this one, and yeah, we're left with two. So we're left with a sulfur and an amine. So I'm going to explain the sulfur first. So the reason this is considered non-polar is because sulfur can't really pull electrons away from the carbon. It's not strong enough, and the carbons are not strong enough to pull electrons away from the sulfur. So it's kind of stuck. It's like this. None of them could produce a charge, and none of them could pull electrons away from each other. So this is the reason why it's considered non-polar, because it's neutral and it's not strong enough to pull any type of electrons around. So this is non-polar, and this amine. The reason this is non-polar is because that nitrogen group, or this amine group, is really stable and doesn't have really any charge onto it. It has a lone pair, but that lone pair is not going anywhere. It's going to stay there, and it's going to be neutral, and it's going to be safe. So for this reason, it's considered non-polar. So just going over, I'm just going to say my tip one more time. If the R groups just have pure hydrocarbons, it's going to be considered non-polar because hydrocarbons cannot have any type of charges onto it. So let me draw a quick question for you guys to demonstrate the point. Let's see, OH. This is NH2. So I'm going to just draw a fake amino acid. It's not real, but for this purpose, I'm just going to use it to explain how to identify non-polar from polar. So we have these two rings attached to an amino acid. So first thing we want to do is find the R group. So this would be the R group since we know how an amino acid looks like, and this is the R group because this is different from a regular amino acid. So this is the R group, and the reason we know this is non-polar because all the carbons, or all the R group, everything in the R group is a hydrocarbon, so it has no charges at all. And going with that logic, it's considered non-polar. So with that reason, this is a non-polar structure, or non-polar amino acid. Alright, off to the polar amino acid. So for polar amino acid, we gotta find something with a charge. So one quick thing to do is find anything that has a charge will usually have an OH or an amino group. So we just find anything with an OH and amino group, and that will have a charge. So this is an amino group, OH, 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 an OH, an OH, amino group, and an amino group. So once again, we're left with two, and I'll explain to you why these are considered polar. So this one has sulfur, and this time sulfur is strong enough to pull electrons away from this hydrogen. So pull electrons away from this hydrogen, it will induce a negative charge. Sulfur is kind of like oxygen in the way. It has the same structure as two lone pairs, and when this hydrogen leaves, it's gonna get the lone pairs from that hydrogen, and it's gonna become a negatively charged sulfur. So for this reason, sulfur is considered a polar molecule, or this amino acid is considered polar, because that sulfur will have a negative charge onto it. And basically, you want to treat the sulfur as an OH group. That's my tip for you. And for the last one, histidine. The reason why this is considered a polar group is because these lone pairs, the nitrogen is pretty negative right now because that double bond close to it, and it has a lone pair attached to it. So what this double bond is going to do, well, not the double bond, these lone pairs are going to do is grab a hydrogen and make this. So it's going to grab a hydrogen and make this nitrogen positive, which is a charge. So for that reason, this is considered polar, because that nitrogen is so negative. It's going to grab a hydrogen and it's going to become polar. So for this reason, this is why histidine is considered a polar group. And this is going to ask one more question before I go on to the next topic. But yeah, that's about it. So how would we, let me draw a random amino acid, or fake amino acid, NH2. For fun, I'm just going to put MCPBA. So yeah, so would this be nonpolar or polar? So MCPA this will be considered a polar because MCPA has an OH group. I know OHs will usually have a negative charge, so it's gonna be polar. That's how you identify it. And yeah, so just to say my tips again, look at the R group and check if it has any hydrocarbons. If it's just pure hydrocarbons, it's considered nonpolar. If it has no charges, it's also nonpolar. If it has an OH or have an amino group, it's considered polar. If it has a charge, it's polar. So yeah, with that, I'm going on what the charges mean and what they do. So nonpolar is considered hydrophobic. Yeah, it's considered hydrophobic. And what that means is when these amino acid group together and make a structure, the structure will look something like this. So this is supposed to be representing a protein. So because it's hydrophobic, we're gonna find all these nonpolar amino acid around the middle. So in this middle part, since it's hydrophobic, we want to get away from the outside and away from water and be inside where it's safe, not touching any water. And for polar, we're going to see the proteins. For protein, we're going to see the outside of the protein is covered in polar amino acid. This is because polar is hydrophilic and loves water. So it's going to be on outside attracted to water. And amino acid with acidic and basic side chains, all that's going to do is affect the pKa of the amino acid. So the way to tell if something is acidic or basic is just check if it has an acidic group. So this has benzoic acid. These two have benzoic acid, and these one have amino groups which are considered to be basic. So these will be basic side chains. All that does is affect the pKa. So if it's acidic, it will have a lower pKa. If it's basic, it will have a high pKa. One of my colleagues is going to explain more onto that in the next video, but I'm just going to touch on the subject. That's all it really does. And onto the last part, I'm going to be talking about the proteins. So I'm only going to talk about 3 proteins from the textbook. So the 3 proteins we have are structural proteins. Structural proteins are just rigid, strong, just keep things stable. And what you'll find them is in your nails, you find them in your hair. They'll find the hair, they'll find the outer shell of crabs or spider or bugs. You basically find them anywhere where you need something strong and rigid. That's what structural proteins do. Transport proteins are just basically anything like hemoglobin, so it'll take one, so transport one from one part of the body to the next part of the body, and just travel easily. And hemoglobin just takes oxygen and delivers all over to our bodies, our tissues, to our organs, and it also takes CO2 back to the lungs. So it's considered transport proteins. And the most important protein of all is enzymes. Enzymes are considered the most important proteins because our whole body is based on reactions, and what enzymes do is they speed up reaction. They lower the amount of energy needed for the reaction to start, and it causes the reaction to happen a lot quicker than it's supposed to happen. So a reaction that could take like a couple of years to happen could happen in a few seconds because enzymes will lower the amount of energy needed for that reaction to happen. And our body's full of reactions, so it's kind of useful for us. So an example of this would be lactase. I'm lactose intolerant. I can't digest milk. So when I drink milk, I have no lactase enzymes ending to break down the lactase, ending to break down the milk, and it will go through my body because nothing's stopping, nothing to break down. But if I take a lactase pill, that's why, and the lactase pills contain enzymes. Those enzymes will break down that lactase while my body can absorb the nutrients. And yeah, that's about all I have for you guys. So remember the tips I said, and you be good identifying between amino acids. Other than that, good luck guys, and thanks for listening.